Good evening, one and all present here. Welcome to today's KAAPI with Curiosity talk. I also welcome the audience who are joining us online. So, I request our director, Dr. B.R. Guruprasad, to formally welcome the gathering.
So it's only formal. Anyway, good afternoon to all of you, and welcome to yet another lecture under the banner KAAPI with Curiosity, jointly organized by ICTS and Jawaharlal Nehru Planetarium. And especially I welcome Miss Claudia Bank for having come here to deliver a talk on fitness landscapes. In fact, when I just read the abstract, really I was wonderstruck by the kind of profound questions which it raises. One is raising the profound questions and dwelling on a new topic, and the second thing is what are all the practical applications it has got. So it is relevant from several points of view. So without taking much of your time, I welcome you all once again to this lecture, and especially welcome Dr. Claudia Bank and for this kind gesture, and I'll request Sharanya to announce the next event here.
Thank you, sir. I request Dr. Prayush from ICTS to introduce the speaker.
Good afternoon, everyone. It is my pleasure to introduce Professor Claudia Bank, who's here with us today from the University of Bern. Professor Bank has had an interesting journey. So she started out reading mathematics at the University of Bielefeld, but soon she moved into population genetics during her PhD work at the University of Veterinary Medicine in Vienna. Following that, she was a postdoctoral researcher at the Ecole Polytechnic, and at the same time she was a Simons Foundation Fellow at the University of California, Berkeley. During this period, I think she focused a lot on using experimental evolutionary data and using that for theoretical understanding of evolution. And after that, she spent 4 years leading a group in evolutionary dynamics at the Gulbenkian Science Institute in Portugal, right before she joined the University of Bern, where she currently leads a group in theoretical ecology and evolution. So let's welcome Professor Claudia Bank again. And just before we start, I would like to extend a small memento as a token of appreciation to Professor Bank.
Oh, thank you very much. Thank you, and hello everyone. Thanks for coming today. And yeah, I'm very excited today to give this lecture. I hope it's going to be interesting for you. And let me switch to start the presentation. Good, so I'm going to talk about fitness landscapes and the predictability of evolution. And so, of course, I'll try to also explain what all of this is. But before I do that, I just want to show you whose work is also represented here. And so this is my research group. The picture is already 1 year old. But I think one of the privileges I have leading this group is to really work with people from many different countries and from different fields. And I think that's what I particularly enjoy, so that we can learn from each other every day and discuss with each other what we find interesting, how we see things, and exchange different kinds of approaches and ideas. And so I'm going to start by introducing a bit to you what we actually do. So in general, we're motivated by what I could call the marvels of evolution. Here are a couple of just exemplary pictures. So how did the diversity that we see arise, the diversity of species and forms that we see? Then, like within the species, how did they manage to get adapted to their environment? This example is a tardigrade. It's also called a water bear, and they have adapted to survive even in space or frozen, or like they are super robust. And how did that happen, and what is the basis of this? And then ultimately, evolution happens on the molecular level through mutations and changes in molecules and protein structures and so on. And we're also looking into that, like what really is this essence of how evolutionary change happens. And then, of course, whereas there are all these wonders of evolution, some of them can also turn into the curses of evolution. Like here, this would be a picture of an influenza virus. And for example, we're studying drug resistance evolution in viruses, but also drug resistance in bacteria, for example, which is also an evolutionary process where genetic changes drive this resistance. And then we can also apply this to cancer, for example, because cancer, yet again, is also an evolutionary process that happens inside us. And so how we do this is by using mathematical models. And I really like this quote, which says, all models are wrong, but some are useful. And it was phrased by the statistician George Borges, and I think it really applies here because we're trying to kind of break down our questions, and we are aware that we can never really capture all the complexities that we see in nature, but we can try to pick those that are interesting and try to figure out what they really do to the process. And yeah, so I already said that, so we really pick out individual factors of interest and to quantify their effects. And today I'm going to talk about one specific factor, which is called epistasis, and I'm going to introduce that in a couple of slides from now. But first I want to just remind you how this thing kind of could work in biology, and this is already a great simplification, but I just wanted to make clear here that we're on the same page regarding the different levels of organization we're thinking about here. So often we start here with some kind of genome. So this would be the sequence of heritable material that is passed on between generations. And so you might have these sequences here, and then sometimes a mutation appears that makes it different. And ultimately, so there again, there's a layer of processes. You get an individual, and it might look a tiny little bit different. So these are sketches of little E. coli, and this one has a black cross on its back, which seems is not very natural, I think. But here this just represents the change that came about due to the change of the genome sequence. And then ultimately we might have populations of these E. coli with crosses or without crosses. And then there might be a difference in how well they survive and how fast they reproduce. And that is kind of the fitness level we're talking about here. And so this is already a very simplified picture. And let me tell you that we're actually even going to simplify more now. So in much of what I'm going to show you over the next couple of minutes, we're going to simplify the sequences even further by making them really short to just a few positions here. So I call these loci. They represent different genes that might be important. And I'll also just use zeros and ones instead of the letters that represent nucleotides. Then on the second level here, I'm going to look at features of the organism. For example, here this could be the division rate, where one of these could have like 3 divisions roughly per hour. The other one has maybe 3.01 divisions per hour. And then ultimately, when we describe fitness, we might just say, oh, okay, we set the fitness of one of these to 1, and then we look at what the fitness of the other could be, and we look at this in a relative framework. So we just say, this is a little bit better. And so sometimes we're even ignoring that middle part. So we just look at things that are described by sequences, and then we look in the end at just their fitness, so their relative difference in how well they reproduce. And to just put the words on this, just to, like get you warmed up here. So this is what I call the genotype. So we're looking at different genotypes. This is what I call phenotypes. And then up here, we talk about relative fitness. And sometimes I'll also mix up these a little bit because sometimes we don't really know what fitness is. So we might just use a phenotype where we think it's very related to fitness and use that as our measure of fitness. So you'll see that over time, and I'll try to also mention it when I do that. Okay, so we'll be talking about models that somehow describe these different levels here and how they relate to each other and how that then affects the process of evolution. So let me introduce to you what then a fitness landscape can really look like. So here again we have like one little genome which we could call like a reference. So this is our starting point, and I'll show you a lot of these plots here where then we have something on the x-axis that just measures how many mutations have accumulated. And then on the y-axis we measure this fitness, the relative fitness. So we can just look at whether something is better or worse. And then we can create these graphs here that connect different genotypes with each other when they have a single mutation between them. So here, this one would be the one that has a mutation at the second locus, and you can see it's doing a tiny little bit better than the original one. The other one is doing much better, in fact, and then we can ask, okay, what happens now if we put those two together? Is that gonna really be just what we expect, namely that these two kind of add up? And that we get like this, you know, kind of additive effect on fitness. And this is where my study topic of epistasis comes in. Because sometimes that might not be the case, right? So it might be the case that when we put those two mutations together, then in fact that is much better than having these two, than we would expect from just measuring the effects of the two. And that could be important in real life when you, for example, think about antimicrobial resistance mutations. So imagine you know these two mutations and they are doing okay, but you bring them together and they give a huge benefit to the bacteria and really overcome the drug pressure. That would be a big problem. On the other hand, it could also be that this combination of mutations is worse than you would expect. And that will mean that we often see that, for example, between different species. So maybe you've heard of these hybrids that you can make between a lion and a tiger. They are called liger. And some of them are really huge and some of them are a bit small. And all of them, they can't reproduce anymore like these hybrids themselves. And that is because one of the reasons why they can't is because the genes are not compatible anymore of the lion and the tiger. And so when you make hybrids, they cannot reproduce because there are these deleterious effects of genes coming together between the genomes.
When you measure the fitness of a genetic variant, in a single cell, you said the rate of reproduction, the phenotype, is the main metric. Mm-hmm. What about multicellular situations?
Yes. So in the end, I think about individuals, right? I'm not talking about cells per se. So really, this is about populations evolving, and then having, like, if you have, okay, going back to this picture from the beginning, you might have a population in which this black cross arises, and because it reproduces a bit faster, it will take over the population. And that is my measure of relative fitness. Okay, so now just some words that are maybe important to know. So here on the x-axis, and I'll show you lots of these plots, that's what we would call the Hamming distance. It's not so important. It mainly just means we're counting the number of mutants here, and this is totally random. It's just one way of representing what I'm going to talk about. Fitness is on the y-axis, and now we already have something that I call a fitness landscape. So the fitness landscape really represents the relationship between different genotypes, those blue things here, and the fitness that we would get. And later on, we're going to ask then what evolution will do, so how evolution will proceed through this fitness landscape. So you can already imagine that if this was our starting genotype, ultimately, maybe this one here would be the one that overtakes the population because it's the fittest one. And that is kind of what we're going to talk about for the next couple of minutes in a little more complicated ways. And so why are we even interested in this? So of course, we're interested in generally like how good mutations can come about. So how large or small adaptive steps are. For example, when you think about climate change, we need to know whether populations will be able to adapt to higher temperatures and so, and that will also, it will also be important to evolve new features and so, and theoretically, if we knew the fitness landscape, we could say a little bit about whether it's possible to actually keep up with the change in the environment, for example. Then one of the things we're also interested in is just measuring in general, like how many mutations are really deleterious, how many are beneficial, how many make pretty much no difference. That is important because also it will determine the evolutionary process. And for example, we know that most mutations are actually deleterious, so harmful for the individual. And so whether or not there are many harmful mutations is also something that we're really interested in, right? And then if we had this landscape, we can also see, okay, how many ways are there actually to adapt? So is this the only way to get much fitter, or are there other ways we could also get fitter? And that's where the prediction comes in, because ideally we would also want to know, okay, so can we actually forecast whether, you know, how, with which mutation antibiotic resistance will evolve? Or is this something that is totally impossible because there are so many different ways of doing it? And then all of this kind of means that in this fitness landscape, all this information about this epistasis, the interaction of mutations, is contained. And so I talk about predictability here, so predicting evolution. But in reality, what we're really doing is to actually first understand whether we can predict, right? So that's why I put the sketch here, because what we're actually, in my group, we're not really working on currently predicting things. Like, for example, other groups, they are working on predicting new influenza strains so that vaccines can be developed. So they really kind of take the sequences and they look at them and they try to make models to predict what the next influenza might look like. What we do is just to think more about, okay, how can we predict, can we even predict evolution, and what is the effect of these gene interactions on whether we can predict it or not. Okay, so now I've shown you a little bit this concept of epistasis, and that we're interested in it from an evolutionary point of view. But what we really do now in my group is we often just sit around and think, right? So that's kind of, I feel like that's what I do most of the day if I'm not responding to emails. So in reality, what we're trying to do is to really come up with good models that describe this process and that we can also then compare with data, and where we can, you know, get a little bit closer to our goal of understanding how this adaptation process works. And I'll show you today also some data, and so I'll just try to guide you a little bit through the thought process that we're doing in our research. And what I would like you to take home from today are a couple of things. Firstly, I'm going to try to prove to you with some examples that epistasis is a real thing, right? So it exists in nature, and there is some evidence that epistasis is there. Then I'm going to show you how we can use models, so mathematical models of these fitness landscapes to see epistasis and to somehow capture epistasis, but in different ways, and I think that's also important. Then I'll show you what epistasis does to evolution, namely that it, and I call this evolutionary, it constrains evolutionary trajectories. And if that doesn't make sense to you yet, please stay tuned, like wait a little bit, because hopefully after this talk, it might make sense. And then finally, I'm going to show you some really just very recent research that we did in the lab, where we constructed a new model and studied this regarding also how organisms interact with each other. And that will be the last part of the talk. Good. So let's start with maybe an easy one, I hope. So I'm trying to convince you that epistasis really exists. And for this, I'm just going to show you a couple of empirical examples. So these are different, like these are mice. And what I'm showing you here is an example of how there are two genes interacting, and there's epistasis. So you can see there's like this one gene called agouti, and these things here, they show you the genotype. So if a mouse has this small a mutations, like so 2 copies of them, because you know these are diploid species, then it will be a black mouse. So basically, if there was no epistasis, we could just look, oh, which genotype does this mouse have, and then we could predict, okay, this mouse is going to be black, because it has 2 copies of the small a allele. But now there's a second locus, which is called, so it's called MC1R, or also the extension locus. And it turns out, well, it also in this picture, it changes the background of the image, but mainly it changes the color of the mouse. And that is basically what's happening here, is that if you have double small genotype, so if you have 2 copies of this allele, no matter whether you had the small a or the big A, you'll always get a light colored mouse. So really, that means that somehow this genotype can, so the MC1R genotype, it suppresses the effect of the agouti genotype, and that means that there is epistasis between those 2 genotypes. Because just by looking at both of them separately, we could not predict what the combination of the 2 does. And so you might ask, okay, so why is this important? And I think in mice, there have been some really cool studies that indeed this, the color of the coat really is very important in some places. So there have been some nice studies showing, so where the researchers, they put little mouse models, so they had these plastic mice, and they were in different colors that resembled the real mice in the places, and they study mice that are really close by in very dark sand and very light sand, and then they later checked how many of the plastic mice had marks from being attacked by birds of prey and so. And from that they could compute what the benefit was of being a dark mouse in a dark place versus not. And so what they found was really that there was a 10% benefit to being a dark mouse. So these genes here that determine your color were really important to make the mouse such that it is adapted to its environment. I'm going to show you a next example, and this comes actually from humans and from malaria, which is a disease that really affects a lot of people every year. And this probably looks very complicated, so I'll try to guide you through this step by step. So firstly, what's shown here are again the different genotypes, and then on the y-axis something called IRR, and that is the incidence rate ratio, and it's a measure of if you get malaria, how bad is it going to be? And in fact, what people have detected before is that some humans, they carry a resistance to malaria, and it can come in 2 ways, and that's where this lower part here comes in. The first thing that genetically you can have is this so-called alpha thalassemia genotype. And so just having that will help you be resistant, basically have less effect when you get malaria. And you can see that because so this ratio here, it compares with like somebody who does not have the resistance genes. So you can see that's why this one here is 1, because these individuals here, they have no resistance gene whatsoever. And then based on which genotype these individuals had, if they have like if they have this kind of combination of genes, then in fact they are much healthier even if they get malaria. And then the same is true if they just have this other thing here, so that would be the reference type. This is one mutant that is called like the sickle cell trait, so that's another way to be resistant to malaria, but only if you don't have the other one, because if you combine the 2, and that's happening here, then in fact it is again very bad for you if you get malaria. So really here we have these 2 genotypes, and they interact such that when you have both of them together, then there is reduced resistance. Okay, so this was maybe a quite complicated picture, and now here I think is another quite cool example. So we have these fish here that live in Mexico called Scyphophorus. And I think this is a very cool evolutionary example because you have this one species which looks like this, and it's kind of, it's living there in Mexico, and then you have another species that has black dots. And it turns out that there is an interaction between a tumor gene and a repressor gene. So that means this repressor gene, it kind of switches off the tumor gene, but it still makes black spots. And so those black spotted fish, they seem to be attractive to the females. And so if these fish, if they live kind of separately, it's totally fine because everyone will have both genes and so they won't get sick. But if they start reproducing with each other and there's hybridization, then ultimately there will be genotypes that just have a copy of this tumor gene, but not a repressor gene, and then they will get cancer. So this is an example where in nature you have 2 species that have different genetic compositions, and when they get together and mix up the genes, there can be a very deleterious effect of this. So these were my 3 examples to just show you that in reality, we do find epistatic interactions. So really they seem to be there, and so I think it's valid to maybe ask, okay, so how do they really matter in evolution? Because just because they are there, that's just, okay, well, nice, we have described this, but what is really the consequence of this? Okay, and so in reality, like when we look at this, people think differently about it. So for example, when we think of molecular biology on this level of proteins or sequences or so, people believe that there is a lot of epistasis because there's, for example, in proteins, there are things that are closer together and further apart. And so they should somehow, everything that's close together should maybe interact with each other. But then in the process of adaptation, there's also some theory that shows that maybe we don't need to think about epistasis. Also, it's complicated, so it's good, so let's just ignore it. But I think that maybe that's not always justified. And then in between species, if you think of these fish, really there are these different mutations where we know they interact and they actually contribute to keeping species apart. And the question now is, okay, is that really something that just happened accidentally, or is this part of becoming new species? And that's what we're going to address with our models. And so in the next part of my talk, I'm going to go now from these empirical examples to introducing to you some ways of creating fitness landscape models. And then we can start to address actually what the consequence is of evolution on fitness landscapes that have epistasis. So this is the part where I'm going to show you different models and how they capture epistasis. And so the first one fits really well into Switzerland because it was developed by Wright, one of the founders of population genetics, in 1930, and he imagined that evolution might be happening on something that looks like a topographic map. So you have hills and valleys, so this would be a mountain here, there would be a valley, and he imagined that real populations, they evolve on these landscapes, so mutations kind of carry the population across this landscape, and overall fitness will probably increase, so beneficial mutations, they carry to up here. And then that's how the evolutionary process might work. But also then there are kind of problems, right? Because maybe this is not the highest mountain in the area, but you could get another one. And I'll get to that in a moment. Because particularly we can think about now, if we have these kinds of landscapes, what are they really going to look like? Are they going to look like really in Switzerland, where you have these mountains with like lots and lots of peaks? Or are they maybe going to look like in Japan, where you have like just this rough Mount Fuji, which overlooks all of it, and there's just a single peak, and the slope is kind of very, very smooth and so? Or are we going to be in the Netherlands, where just there's no peaks whatsoever, and everything is the same, and there's no way to kind of climb the hill at all? And so that's exactly what the different models somehow also contain in themselves. So Wright was definitely was convinced that we were more on the Switzerland side of things, and that there might be many peaks in these fitness landscapes. And then, of course, there's immediately a problem, right? If populations try to kind of get better and better at reproduction, and they climb a peak, how are they going to get from there to an even better peak? Because somehow they might get stuck, right? So you can immediately imagine that if you have a fitness landscape like that, you might get stuck. And that's why he needed other forces of evolution in his theory to be able to get from one peak to another. And he believed that, for example, you could get local populations that then where you get, migrants, and then they create new diversity, and that would be how you could get from peak to peak, especially if population size also changes. So quite complicated process. Where not just selection is really playing a role in getting you up to that mountain in Switzerland. Then, around the same time, Fisher's geometric model was proposed. And here, this is also, you can again see kind of a space here. It is two-dimensional. And here, basically, we're not thinking about genotype space, but we're thinking about phenotypes. So it could be like color here, population, like size of individuals here. And then it could be that there's like one optimum. So this is our rough Mount Fuji. Our Mount Fuji is kind of coming out of the projector here, right? And so if you're somewhere on this landscape, you want to climb up the hill, you might get mutations that are randomly just going in any direction. And so it's particularly good if you hit one of these ones, right? So this arrow here, it takes the population closer to the peak, and so it will be a beneficial mutation. It would be pretty bad if you got this mutation, because it actually takes you further away, so you'll be down in the valley and you have a deleterious mutation. And again, if you have this kind of model, you can immediately imagine more about the evolutionary process, namely that as you get closer to the optimum, and that is actually shown here, so you might start really far away. And then eventually you get closer, and now there are less of the arrows that are beneficial because you're already so close, right? And so immediately when you have a model of this, like this, you can conclude that you expect that adaptation should get slower and slower as you approach this adaptive peak. So mutation effects, beneficial mutations are not so beneficial anymore later during the adaptive process. And then finally, I'm going to show you this model here, which is really sketched very roughly, where it's more like a Swiss cheese landscape, right? So it's all very flat, like the Netherlands, but with a couple of holes. And these holes, they represent this epistasis where there might be overall very little fitness increase, but sometimes you might hit this Liger phenomenon where different mutations don't work well together and they make populations unfit. And in that case, you can immediately, if you think of this as a model, then you might believe that, okay, we don't really need fitness increase over time. We could just get neutral evolution to happen, and still there could be eventually some bad effects on hybrids, just because populations have gone to different places, and when they come back together, they have these epistatic incompatibilities. Okay, so I think that what I'm really trying to show to you here is that we can make these different models, and it's important to notice that then this model will always reflect the philosophy of the modeler, right? So really what you believe about evolution also goes into the model, and then maybe it will also affect what you get out of it, right? And you can see this here because, for example, Wright, he was in fact almost like the inventor of the so-called genetic drift. So he was a person who thought like, oh, randomness is really important in evolution, and this exchange of migrants rather than selection maybe. And so that's why he believed that this map must be very rugged, and then like these other factors play a strong role. And in his age, he was having all these fights with Fisher because he believed something completely different. He believed that populations are huge and that there really is like selection is extremely efficient, and that's why he thought that especially locally there would always be any way of kind of increasing fitness is somehow possible, and we don't need to care about like additional fitness peaks or anything. And then on the third point here, Gabrielets, when he proposed this holy landscape model, he wasn't thinking about this short-term adaptation, but he was thinking about species, so maybe millions of years of evolution where these small selective steps, even if there are many, they might not really play a role, but where evolution is now, we're really looking at these big effects when hybrids are formed. So that the time scale he was also thinking about was a very different one. Okay, so now you might ask, okay, so yeah, now she's shown us these like really theoretical concepts, but what do real fitness landscapes actually look like? And the good thing is nowadays we can measure fitness landscapes, and they're increasingly large fitness landscapes. And so a couple of years ago, I got involved in some studies of fitness landscapes where the experiments were performed by a PhD student in our collaborator's lab. And he performed experiments on this here. So this is a picture of a protein, which is a heat shock protein in yeast. So, so basically it's just the structure of this protein. And here you have like a tiny little area which has just a very few positions in the sequence that we could, however, then mutate and we could see, okay, what effect does each of the mutations in this small area of this protein have, and how does that then affect the individuals and the fitness of these individuals. So really tiny changes, could they really have, like, what would be the effects of making tiny changes in this protein on the organism? And so one study, for example, that we did was to make double mutations. So basically, instead of just looking at single effects of single mutations, we would just make double mutations, compare them with the single ones, and measure epistasis. And there we already saw that, whoa, there was a lot of epistasis where putting two mutations together really resulted in much lower fitness than we expected by having just the single mutations in this genome.
Is there an intuitive way of understanding why this is? Why one is 47%, one is only 0.7%?
Yes, so I guess, so I took out this picture, but in general, I think that one of the common findings is that especially within this protein, if you do too many mutations, you're going to destroy the function of the protein. So sometimes you might find something that works well, but then as the more mutations you make, the more likely you're just breaking the structure and so. And so there should be, and I've showed you this also in my talk, there's usually this pattern where sometimes you can find something cool also a few steps away, but most of the time there's like a lot of deleterious genotypes created if you add more and more mutations. Okay, and in fact, I'm going to show you this here. So this is another fitness landscape. You can see this is like pretty much like some of the pictures I've shown you with this Hamming distance and the fitness, except that now like each of these, you can't see the genotypes anymore. And so there we had 600 something genotypes. And again, these were the single mutants, and there's no predictability whatsoever, right? So what happens if you add more of them together? And in fact, what we found was that there was a lot of negative epistasis, so mutation combinations were kind of bad when we added more and more mutations. And then there was also some positive epistasis, because trying to predict the highest fitness we would get from adding many together was also totally impossible. So both of these kinds of epistasis played a role, and that really suggests that just looking at this one step was not a good, it's not a good idea if we want to learn a bit more about the bigger landscape. And in the meantime now, people make these experiments with much, much, much bigger data sets. So here is this is one, probably hundreds of thousands of genotypes, and still these give you very similar patterns. And people have now made much progress also characterizing this with how general this is. And one of these phenomena that you were referring to, it's called kind of a global epistasis, where overall that seems to be happening. Okay, so now I've shown you that we have models, and then maybe there's also some kind of ruggedness in these fitness landscapes, but still we don't have any evolution happening here, right? So there was no movement or anything through the landscape. And that's what I'm going to show you next. I'm going to show you one more model, and then use this model to simulate evolution in the computer. And that tells us really something about then predictability and repeatability of evolution if there is a fitness landscape like that. Okay, and that's going to show maybe the point I find most important, namely that epistasis constrains evolutionary trajectory, and that means it makes some ways possible and it makes other ways impossible. And you'll hopefully see that in some of the next slides. So for this, we create a computational model. So basically, we write some computer code, and the code can compute a fitness landscape for us. And then we pretend that evolution happens in this landscape. So basically, we start somewhere down here, and then we say, oh, mutations can happen. Where will we go next? And we do that successively until we reach some kind of fitness peak. So until we've reached the top of our mountain. And this model, which I'm using for this here, is called the rough Mount Fuji model, because in fact, it allows you to have landscapes without any epistasis, and then you can tune them in the computer such that you can get any level of epistasis until you reach a completely random fitness landscape. So in this one extreme case here, each mutation effect really predicts the next effect. So if you look at this one here, it's a one mutation at the first position, and if it happens, for example, here, it has the exact same effect, right? So these lines are just parallel, and so that means there's no epistasis in the landscape, and everything is kind of nice and additive and so on. Whereas in this extreme case here, we just draw a random value for each of the genotypes, so that becomes extremely like rugged. And in fact, that means that here we have a predictability of just knowing the single mutation effects allows us to predict every genotype's fitness. So there's a lot of predictability here. And then on the other hand, here we can predict anything, right? Because every value is random. And so that means we can't really say anything except if we have measured really every possible genotype. And there's kind of, so now our model allows us to find anything in between also. And I'll show you how we can use simulations now to understand better what consequences this would have for evolution. And so what it does is, here now I'm showing you bigger landscapes so that it gets a bit more interesting. So now instead of just having 3 loci, I have 10, but otherwise it's exactly the same thing. And now the gray lines would be just your genotypes in the landscape, just like before. And whenever it's blue, it means that evolution can actually go there. So we start a simulation here in this lowest fitness type, and we run the simulation until it reaches an endpoint. So we can't increase our fitness anymore through mutation and selection. And so clearly there are a couple of things that are kind of intuitive from this model, right? So firstly, of course, you can look at the number of fitness peaks, and as the fitness landscape gets more rugged, there's more epistasis, and so the number of fitness peaks increases. So that, I think, you probably just got from how I explained the model, right? Because we're going from this one extreme case to the other extreme case. Now it gets a bit more interesting, because evolution kind of happens in this landscape, and in fact what happens is that if we think about how many different solutions we can get, so how many ways there would be to kind of increase your fitness and get to a peak, then that is maximized for intermediate epistasis. And that's because here it's so rugged that you can't even go far anymore. You just kind of get stuck in a peak very near you and not really explore all the good solutions. And then now, if we think about repeatability of evolution, again, it's kind of funny because now in this, whereas in this additive landscape here, you have only one peak. Obviously, you have to go there all the time. There's no other way, right? But you can take any possible paths upwards, right? So there's a lot more variety of different paths to get to the global peak. And this decreases again with the amount of ruggedness in the landscape, where in the end here, there are only very few mutational steps that are actually being taken in the landscape when you simulate evolution. And now if we think about how new species might arise, right? How do we get this diversity of like lions and tigers, for example? We can think about now evolution, let's say, happening on 2 islands for like 1 million years. And then we can check in which of these landscapes, how far apart those genomes would be after evolution. And that's shown here basically. So now if we run the simulation many times and we compare how far apart are the final sequences, then again, this is the highest for these intermediate epistasis landscapes. And that's because here there are a lot of peaks already, but they are kind of further apart, and the landscape is explored by evolution such that in the end we get like a higher divergence between different simulations than before. So maybe if we think about this now from like a biological point of view, now, if we have this intermediate kind of epistasis, that would mean that this is kind of maybe the best case for getting speciation to happen to very different kinds of genomes.
Yeah, but suppose I increase the population size, would this last result still hold? That means you will get more trajectories with intermediate epistasis?
So of course I made a very short explanation here. So we're talking about allopatric cases, right? So we really put the population into two different islands and let it evolve. And in fact, we're assuming strong selection, weak mutations, so the population size does not matter here. Huh? Yes, I don't know. Yes, interesting question.
Could you give an example of something that is one Hamming distance away? Like, I want to understand what it means when...
Yes, so it's really just a number of mutations that separates two sequences. You were talking about the Hamming distance?
Yes.
So really, if you have, like here, I think I had this, yeah. So basically, this one was our 000 type, right? And these are all the types that have just one mutation. So they just are one step away. And then the next one, all of them have two mutations as compared with the first one. And that's what we mean by the Hamming distance.
So how far apart would be, say, two different species?
Yeah, that is a very good point. So yeah, I think that's actually an interesting, difficult question. So yeah, so if we think about, okay, I might be wrong. But I think that if you compare, for example, humans and chimpanzees, there is like a 2% of the genome is kind of different. So this doesn't give you now a number, but they have like huge genomes. And so 2% of these huge genomes are kind of different. And yeah, I guess that doesn't completely answer your question. Yeah, I don't think I can put numbers on it. But I mean, maybe later we can look it up, how large these genomes are, and then you would roughly know.
Madam, you are mentioning fitness. In fitness in what sense? You're talking phenotypes and xenotype. Fitness in the what side? What sense? Either in the height or weight or something like that?
That is a great question. And in fact, we often use different kinds of measures of fitness. So in these fitness landscapes that I showed, in the empirical ones, we just compare the growth. So basically, we put the yeast cells together in a culture, and we see which one grows faster than the other, and so we compare this relative fitness. And in other cases, for example, we might just kind of measure their growth rate or the division rate or something. And choosing a good measure for fitness is a very difficult problem. But in general, our fitness is really just some kind of advantage in reproduction over another one, like relatively. And here, since the good thing about modeling it is we don't really need to decide, right? We just say, okay, we're talking about this abstract concept of something that makes you reproduce faster than before. Okay, so now getting back to the predictability. This here means that if we have an additive fitness landscape, we have predictability of the endpoint of evolution, right? Because only one is possible. But we cannot predict at all how this is going to happen and which sequence of mutations will actually occur to get to this point. On the other hand, here, if we look at it, if there's only a few paths that evolution can take, so probably these will be taken many, many times, and that implies that if you've already seen this a couple of times, you know roughly which mutation is going to happen, because these are the only ones that can really lead to a higher peak. And now again, you might be asking, okay, is there any evidence of this kind of thing happening in nature? And for this, I've brought another example, and this is from influenza evolution. So in lab in the US, they studied how the human influenza genome has evolved over 50, 60 years. So nowadays, you can basically keep track of the sequences. I mean, probably many of you know that for COVID now, there are all these sequences, but also for influenza, there's a lot known about how the sequence changed over the last 50 or 60 years. And so what they did was they looked at these sequences, and then in the lab, they could recreate some of the mutations that happened during the evolution of the virus in nature, right? And now in the lab, they tested what the effect of different mutations were and whether the order of mutations mattered and whether there was epistasis. So what you see here is basically time, like so this was the sequence of mutations that happened in one protein of, one gene of influenza. And then on the y-axis is again a funny measure of fitness, getting back to your question, which is the melting temperature of this protein. And so why does this matter? So in fact, it's an indicator of the stability of the protein. And if it has very low melting temperature, that means that in that case, the activity of the protein is reduced and the growth is also reduced. So this means that probably if the melting temperature was so low here, then the virus could not survive. And so what they found was that there were a couple of cases here, like this one, for example, where there was a combination of mutations that together really led to a good virus that in fact attacked the human immune system more efficiently than before. So this is not shown here in this plot. But really this mutation, which was the one that was good to evade the immune system, could not have occurred just on the previous background, right? So then it would have led to a totally destabilized protein. So what they found was that they found these mutations which were involved in the human immune response to the virus. But then they had basically these epistatic cases where there was first the protein had to be stabilized to evolution, and only then the immune evasion mutant could appear. So here really like epistasis caused that, like because of this epistasis, first this mutation had to appear and that one, because the other way around it wouldn't have been possible. And that is really, I think, a cool example of what I showed you in my model also, that there is this constraint that comes through epistasis that not every mutational path is possible. And so another interesting aspect of this was that they studied this in humans. Oh yes, there's a question.
You go back to, this is for an human influenza virus, right? So this is a virus. COVID was also a virus. How well can we exportate from here to a complex life form or a multicellular life form? Because virus at the end of the day is a single cell, DNA and RNA.
Yeah.
So how well can we say with what we know from this to the other complex life forms? Can we exploitate that?
Yeah,
too?
I really appreciate your question. So the good thing is, I mean, the nice thing why we have so many data like this is because it's a lot easier now to go with influenza in the lab and test things like this. Well, but I mean, there are examples like this also from higher organisms. For example, I don't have pictures of this. There was a case in monarch butterflies, these beautiful big butterflies, and they found that they had, like, as compared with an ancestor, they had toxin resistance to a toxin. And this resistance was, you needed 3 mutations for this resistance. And they figured then out that basically when they compared across a lot of different insects, that always the same order of mutations had appeared to get to this toxin resistance. And I wonder if they could test this in the lab already, but just this kind of seems to be some evidence that not every way is possible to get to this toxin resistance, but you really need this specific order of mutations, so there was most likely epistasis there. So there are a couple of examples. It's just that I think that currently the technically it's kind of much easier to do this kind of proof here in single cell organisms or viruses. Okay, so basically here this was our fitness measure. We have this evasion of the immune system, which is only enabled, through first stabilizing the protein. And so they found this in human influenza, and what I found quite cool was then they also studied the same protein in swine influenza. And of course, so if you think about the virus lifestyle in humans versus swine, so humans usually they get infected several times in their lives, right, by influenza. And so probably the immune system of humans is kind of prepared and so has evolved to really react to this virus. And really the virus needs to adapt to the immune system all the time. Whereas in swine, usually they get only once infected due to their shorter lives, they only get infected once. And so when they did the same study trying to find these epistatic trajectories in swine, they did not find them. And so I think that raised a very interesting question, namely whether there could be kind of how much then this epistasis is also changing across different environments, because I mean for the virus the environment is the host, right? And so apparently the virus in humans, a influenza virus that affects humans experiences a very different environment than an influenza virus in swine. Good, so I've shown you now, I hope I convinced you a little bit of this effect that epistasis has in constraining evolutionary trajectories and enabling some evolutionary paths and not others. And now the question is, how many more minutes should I go? Okay, yes. So now comes maybe the most complex but also last part of my talk. Where this is about what we've really we had this paper just accepted a few days ago, so this is like fresh research where we're trying to use fitness landscape theory and going from this very static picture that I've shown you of this fitness landscape to a case where the fitness landscape can change all the time, because in the population there may be individuals that interact and they might compete, for example, for resources. For example, you know, in bacteria they might eat different types of sugars, and some bacteria might be better at eating like one sugar, and the other one then might be better at the other. And then when mutations happen, this changes the competition of the different bacteria. And so this means that maybe we shouldn't even look at this landscape like at a topographic fitness landscape, but maybe it's more like a seascape, right? So where actually all the time the fitness landscape is changing, and that is really what evolution then experiences over time. And I think we're only at the beginning of developing different kinds of models to really address this and understand how epistasis changes across environments and what kind of effects that has. Good, so going back just to remind you, so I was talking often about genotype to fitness maps, right? Where we somehow put this into one category here. And now in our model, we made it explicit that we would go from a genotype to a phenotype and then to fitness. And here, just to kind of guide you a little bit smoothly into it, remember this very first genotype landscape. And now we're just going to turn it around 90 degrees and plot it over there. So this is the same thing. Here we have now a genotype mapping to something like a phenotype or a trait. So that could be how good you are at eating glucose, for example. So this would be our fitness landscape. So different genotypes have different abilities to consume some kind of resource. And now, if you remember this model here, that was a phenotype fitness landscape. And then we're also going to put on top, that's where it's got, see, it gets so complicated that the pointer doesn't even like it. So what we can do now is to map genotype to phenotype. And then we have another mapping, like here, this, for example, in one dimension, this would translate to this here, where we would just say, this one ends up here, this one ends up here. And now we can go back and just see, okay, what did this look like just from the genotype to the fitness level? You don't need to, memorize all of this. I'm just trying to make a point that we can make models that include these different levels. And so we have both a genotype-phenotype map and a phenotype-fitness map. And so in this new model that we developed, we basically had something where this part here became a changing thing. And that was because there's resource competition between individuals where mutations might lead to changes in how well individuals can eat resources, and then it changes also how everyone else, kind of how many resources are still there for everyone else. So for the details, you can read the paper. But what this then really looks like, so basically we use this model that I introduced to you for this part, and then we have this resource consumer model on this part, and we get fitness landscapes that change, and on this we simulate evolution again with new mutation and like chance events and so on. And I'll just show you what it looks like, namely like this. So now here we have the different types of mutants again, so this would be our 0, 1, 2, 3, 4, or 5-step mutants, so the Hamming distance again. And fitness is on this axis, and then the more red point is, this would mean that now all individuals are this type, and now over time you'll see how the frequencies will change, mutations appear, and the fitness landscape changes. And that happens because the different individuals, they have different ways of consuming the resources, and so they compete with each other, and that changes the landscape altogether. And so that's how we get this dynamically changing landscape where still they try to climb up, but they don't get immediately there, right? There's epistasis, there are these interactions, and after some time they kind of get higher, but it takes a while. And then ultimately there will still, at the end, there will still be fluctuations in the landscape just because there's new mutation coming in and it mutates back, and then there's a bit of, you know, kind of chance events and so on. So here now you can see that towards the end there's a couple of genotypes that are maintained in the population at fluctuating frequencies. And so what we did with this model then was to try to figure out, okay, what does epistasis do? Is it really helping here? Does it help this diversification process? Because often that's what we think about, right? Like, what really creates this diversity that we see and maintains it? And in this case here now, where we already have ecological interactions, is epistasis good to have even more diversity, or is it maybe even bad? And so, of course, since I always think about, oh, epistasis kind of makes fitness peaks, I thought, probably we will get more diversity when we have epistasis, right? And I'll show you just, I'll skip this one, but here now you can see here epistasis on the x-axis, and then the colors, they show you two different types of diversity. One would be genetic diversity, haplotype diversity, and another one would be phenotypic diversity, how different are different individuals at consuming the resources. So you can ignore this axis, but what you will basically see is that what we saw was contrary to what we expected, that epistasis was in fact not good for having more diversity in this model here. However, it seemed that there was some effect that some epistasis could be good to create very phenotypically different individuals in the model over the long term. And to us that was quite surprising, and now we're trying to actually go a bit more into this and develop like more mathematical models also to characterize this process here. So basically now, oh, there's a question again.
So have we figured out on the first graph, why does epistasis actually reduce the diversity? Mm-hmm. Have you ever figured it out, or are we still in the process of figuring that out?
Mm-hmm. So I cannot tell you for sure. We're trying to understand it right now, and that's why we're also getting into the math, because that's where really the mathematical model can help us, right? Because it can give us some more general results. And so I think it is because overall, if you have epistasis, it's not as like straightforward to go through the landscape, and maybe there are not so many options to go to like diverse peaks, and so that might be the factor that reduces diversity, but I would not take bets on this right now. Good. Oh, another question.
In case of the graph, is it assuming neutral evolution or is it assuming selection?
Is what?
Is it assuming neutral evolution or is it assuming selection?
Yes, so it has, I mean, this is basically the strength of selection, But in this model here, we also assumed like a finite population size, so there's also a little bit of genetic drift, if you're familiar with that. So it's kind of a combination of selection forces, but also a little bit of randomness in there, which is probably a bit more realistic than some of the other models we have. But it also makes the analysis more complicated. So we might, for example, for the next model, just go to a simpler case again, where we ignore randomness and just focus on selection. Cool, good. So this one I skipped over. So in this last part now, I've shown you basically how things get even, it was already a bit complicated to think about epistasis, and now we made it maybe even a bit more complicated by creating this changing landscapes. But what we're hoping is to get some kind of understanding from this about how this really changes evolutionary dynamics, and when this then we can also start thinking about when this might matter, right? Like, do we really need to consider this whenever we study evolution, or is it still fine? Like, like here, for example, if we just look at adaptation over like some time scale, is it maybe still fine to ignore epistasis? So this leads me to my wrapping up here by saying, or reminding you or hoping that maybe I've made some points about how there is epistasis, so there is interaction of genetic variants. We can make fitness landscape models, and we should also be aware that then these will always be a little bit influenced by how we think about evolution. Epistasis, what it does is it closes some potential evolutionary paths, and we're not yet sure whether it can help or hinder ecological diversification. And so getting back to this, so yeah, I think that I could not provide the total answer to what the role of epistasis is yet, but I hope that I also made you a bit curious about how gene interactions can affect evolution on these different levels, ranging from the protein to the species to the diversity, to general kind of ecosystem diversity levels. And so with this, I'm also going back to my acknowledgements, because as I said, this work is driven also through interactions, but mostly positive interactions with collaborators who also give us data to analyze, who help us define good questions, and also my team, which is amazing at doing research, and specifically that last project was mainly driven by a former postdoc, Andre Amado, and so thank you very much, and I'm happy to take more questions.
Hello, nice talk. So there was one graph of fitness where there were some numbers, 0.1, 0.2, 0.3. So what do they actually represent, and is there any higher scale of the fitness which tells something about the thing?
Yeah, that's a great question. In this talk here, mainly they did not represent anything meaningful except that something is better or worse than another thing. So you could not, of course we can, often when we measure phenotypes, so you saw the melting temperature, that was a real measure, but we really were only thinking about percentages basically. And so it's not some meaningful number where you could say if it's 1.3, that's 1.3 divisions more or something. It's really just a relative scale here.
So about the melting temperatures, I don't know, I may be wrong, but I feel like we get more sick with cold during winters and not in summers. And summers in India could go as high as 45, 47 degrees Celsius. So is there a correlation between this and that?
So I think that's also a great question. I mean, I think that for these kinds of things, there again, ecology is also super important. So basically, how does the virus live? And in fact, what viruses don't like is when it's dry. They really like it when it's they like it when the air is dry, and so they can kind of get better into your nose. And so that's why they tend to infect us in the winter. And whereas then in the summer, so at least that's kind of yes. So here it's more about how the virus really likes to live, which is again a whole different story. But I think that actually if you think of different viruses that give you different kinds of colds, I think there are even studies showing now, because there's a lot more tracing of this, that actually some of them are a bit differently seasonal. So you get kind of, at some point you get flu to be more prevalent, and then a little bit later you get another one to be more prevalent. And of course that will also reflect the adaptation of the viruses to different kinds of scenarios, and also the competition, right? Because also the viruses will compete for, infecting us. And so probably it's a combination of genetic adaptation to different environments and also the possibility to infect more humans at a certain time.
So regarding the negative impact of epistasis, so I have something in my mind which I just wanted to tell you, and just you tell me if that is correct or wrong. So we all know that there were some species which got extinct, right? So can we say that due to the change in environment, there are some mutations which impact negatively on the creature and make it like die or maybe get extinct, so that it cannot survive in the upcoming time, like because of the some changes in the atmosphere? Can we say like that?
I think I really like the question. However, I like to put it a bit like some million years forward into nowadays time. So what people try to do sometimes is nowadays what they call genetic rescue. So when a population is getting close to extinction, they sometimes try to cross in sometimes even a different but slight close species to rescue that population, because they know otherwise it will go extinct. There's no genetic diversity left. And so I think here that we get exactly to your question, because if there was some incompatibility between those species, actually it might be even bad to cross these two species, because then you get the negative effects. But what is also observed, and you see this in plant breeding, for example, is that sometimes you have lineages that are a little bit apart, and you get positive effects of epistasis. So it's called heterosis. And so, for example, some of our crops are really based on crossing different species, and that is good. And so I think one of the really big questions is, so people in plant breeding, they really study a lot of this positive effects and this positive epistasis, and people who study animals, they often look for incompatibilities, but I don't think that these exclude each other. And I think one of the questions is really, so how much is there of each? And if we now, for example, wanted to rescue a species, we would really need to know, okay, is this going to be a good thing or a bad thing? Can we just kind of give the population some new genetic diversity, or might we even drive it to extinction because of some negative interactions? So yeah, so I think it's a quite timely question.
I wanted to ask, the number of fitness peaks, is it a random number, or is it like for a particular process there's a higher number, like a set number?
Yeah, this is a cool question too. So those models that this Raffman-Fuji model which I presented, it's a probabilistic model. So you can, the disadvantage, I'll start with that one, is that it's not motivated by like a mechanism or so. It's only based on some small, some statistical parameters. And so every time you generate new fitness landscapes, they look a bit different, and they might have a slightly different number of peaks. And so basically, obviously, so if it's additive, there's only one, that's easy. And then it goes to a high number for the random landscapes. And I think there has been some math showing what the actual limit is and what the mean would be, maybe for any of these values. But basically when you generate a new fitness landscape in the computer, it might always have a slightly different number of peaks.
Just continuing on what the gentleman had asked, you had said that we can actually model it and see that when it can get extinct. Can we apply that modeling to have the similar effect in real world? So for example, let's assume that we want to model, say, the extinction of mosquitoes, or we want to model the extinction of influenza. Is it possible to do with this and say that, okay, these two mutations or these set of mutations will lead to its eventual that? Is it possible to do such, and is it possible to implement that in or make that happen?
Yeah, I think that yes, there are lots of models like this. And what I referred to you earlier is called, like, from the theoretical concept, called evolutionary rescue. And however, I think that epistasis is not maybe considered there so much, because it's already quite complicated to do it without epistasis. And then I think one of the very important factors when we think about higher organisms is just the genetic diversity. And that might help you predict a little, like, get towards predicting whether a population can persist or not. So that's the first part. And then the second part is how can you actually maybe rescue the population. But of course, I think another application really is also viruses, where people try to model how can we kill the virus, because there we want the opposite, right? We do not want it to get rescued by adaptation. And there, for example, we did some models addressing, so you may have heard about these mutagenic treatments that were developed and sometimes also used against COVID. So where a drug was given that increases the mutation rate of the virus, which sounds counterintuitive at first, but because most mutations are bad, basically that will kill the population because the genome is just eroded due to these deleterious mutations. And there we did some models where we showed that certain types of epistatic mutations might be quite dangerous for drug resistance adaptation well, we kind of applied it more or less to influenza, but it could apply to different kinds of viruses.
These models that we make for epistasis with respect to fitness landscapes, can we apply them to macroevolutionary scales as well, or is it only at microevolutionary scale?
Well, I mean, maybe, so I don't know if so in academia, we need to write grants to get money to do things. And so ask me again in 2 months, because I tried to write a grant about this, and we'll see if I could convince the reviewers who decide whether I will get the money, whether it's something that we can study or not. So let's see.
If there are no further questions, there is one question.
I'm not sure if this question would make sense, but are we looking at how two separate genes are undergoing epistasis and, say, another set of genes undergoing epistasis? Do they also interact, or is there something that's happening like that?
I think that is a great question. And so I was teaching at the ICTS the last week, and one of the questions that I also tried the students to think about was, now, I mean, we know that there is some epistasis within genes, we know there is some epistasis between genes, but how much is there really? And how, like, where does it matter and where doesn't it matter? I don't think we know that really yet, because most of the studies are really case studies. And so I don't think we have a totally, like, a general good prediction yet where we would, how much there really is. And so both, I think, though, that people are really interested in figuring this out better. And also on the modeling side, there have been some quite cool, new papers that are a bit less artificial than the one that I showed you.
Another question. You had mentioned you were using some probabilistic methods to build up those maps. Do those methods have inherent biases, or are they very non-informative probabilistic methods?
So I mean, here we're just using this probabilistic model to simulate, to get to the landscape. And I think the main criticism I have against these models is that they are so unrealistic, right? Because real epistasis will have some kind of mechanistic reason. For example, these interactions within the protein, or also, I don't know if you're familiar, but there are these, like, developmental pathways where genes interact, and there are these cascades of genes. And obviously, that's kind of where you would look for epistasis first, because changing one may have an effect on the how another gene works and which kinds of signals it receives. And so I think that rather than thinking about like biases or so, I think, yeah, I guess the biggest problem here is that what we create here are very, people call this like regular fitness landscapes because they have the same rules underlying the whole space, even though it's rugged, so it looks kind of not homogeneous, but it's created through very homogeneous rules, and that seems to be something that real epistasis is not like. It's more like modular, so you have kind of like within genes, and then like within pathways, and then with these kinds of modules, I think then you would get to a more realistic type of picture of landscapes.
So, when we say epistasis is interaction of genes, do we mean inside an organism or between organisms?
Another great question, which I also kind of touched upon in my teaching last week, because of course these are different levels. So one of the things is that just we could hypothetically make mutations and see if they're epistatic, or we can go, for example, to the fish and see, are there actual epistatic alleles that are between species? And these are two different things, because one is kind of the substrate that evolution can work with in its process, and one is like the consequence of evolution. And so that's again also leading back to this, to my slide, which says like we still don't know, for example, in the process, so going from the substrate to the consequence, is epistasis just kind of when you have, for example, this incompatibility, is it just a side product, or is it kind of important in the evolutionary process to create different species and so, yeah.
So also, what does it mean when we say genes are interacting? Like they are part of a DNA sequence. So is it like, are they physically interacting? What does it mean when we say genes interact?
Yes, different ways. I mean, in the case of the protein, often it might be that so you have the sequence, and then it's turned into amino acid sequences. Ultimately, they're kind of wrangled into a protein, and so here you have certain residues that touch each other, and so there's for example an example where they found that probably just by changing a little bit changing two to kind of relate to things that bind to each other, you could get you could drive a cool change in the protein that was important, for example, in mammals or so. And there it would be within the gene and would really be almost like a physical interaction. But then if we think about these pathways, often it's that one protein produces something that another protein then uses, or that triggers the production of some enzymes or so. So there is more of a more kind of substrate interaction maybe. And yes, so I think that there are different ways in which it can happen.
All right, if there are no further questions, let's have a huge round of applause for the speaker again. There's tea and coffee outside. If you have more questions, you want to interact with the speaker, please feel free to have that. The next KWK talk is on the 17th of March right here. And there are other talks that are also happening. One is on the 10th March, 11th March in ICTS. You can register in our website and come for the talk at ICTS. Thank you very much.
And thank you all for coming and for the cool questions that you asked.